Assalamu alaikum wa rahmatullahi wa barakatuh. We will continue together the endocrine system, and today's lecture will be the pancreas. The pancreas. Pancreas. Pancreas is the second largest gland of the body after the first largest gland, which is the liver. So the pancreas is the second largest gland of the body. Pancreas considered as a mixed gland. Mixed gland is its functioning part, which called parenchyma, formed of exocrine part and endocrine part. Pancreas considered as a parenchymatous organ. Pancreas is a parenchymatous gland or parenchymatous organ, so it's formed of stroma and parenchyma. These stroma, which form the external and internal shape of the gland, and the parenchyma, which contain the functioning cells that secrete enzymes and hormones. Stroma of pancreas formed of capsule, trabeculae, and reticular network. Capsule of pancreas formed of thin, delicate connective tissue. Thin, delicate connective tissue. And the trabeculae are thin, delicate connective tissue which descend from the capsule to divide the gland into distant lobes and lobules. The trabeculae descend from the capsule to divide the gland into lobes and lobules. Then reticular network. Reticular network to form the background of the gland, but not stained by hematoxin and UC. It can be stained with silver to appear brown in color. Then parenchyma of the pancreas formed of exocrine part and endocrine part. The exocrine part formed of acini and ducts. Acini and ducts to secrete hormones in these ducts to reach the duodenum to help in digestion. And the endocrine part formed of islets of Langerhans. Islets of Langerhans which secrete hormones directly into the blood through fenestrated blood capillaries. This diagram to illustrate the parts of the parenchyma of pancreas. Parenchyma or functioning part of pancreas formed of exocrine part and endocrine part. The exocrine part, as you studied in the last year, the exocrine part formed of secretory units and duct system to take the secretion. Secretory units called pancreatic acini. Pancreatic acini. These pancreatic acini release digestive enzymes as polypeptidase, amylase, lipase, nucleotidase, all these digestive enzymes to help in digestion. So it's secreted through this duct system directly to the GIT or the duodenum part of GIT. And the endocrine part formed of islets of Langerhans, called the islets of Langerhans. These islets of Langerhans scattered are pale areas scattered between the pancreatic acini. Multiple pale areas. Its number its number is about 1 million in the pancreas and more numerous in the tail region of pancreas. These islets, called the islets, as it's scattered between pancreatic acini. These islets of Langerhans is the endocrine part, formed of group of cells that and fenestrated blood capillaries. These cells secrete hormones as glucagon and insulin hormones directly in the fenestrated blood capillaries to reach blood circulation. So, the pancreas is a mixed gland formed of exocrine part and endocrine part. Islets of Langerhans, which is the endocrine part of pancreas, form about 1 to 2% of the volume of pancreas. In human pancreas, islets of Langerhans is about 1 million, mainly in the tail region of pancreas, Structure of islets of Langerhans, it's formed of cords of cells separated by fenestrated blood capillaries. This diagram showing the pancreas as it's formed of exocrine part form of the pancreatic acini, which secrete pancreatic enzymes through ducts to reach the duodenum, and the endocrine part, which are pale areas called the islets of Langerhans, scattered between the pancreatic acini. These islets of Langerhans scattered between the pancreatic acini and contain fenestrated capillaries to release its hormones directly into these capillaries or to reach the blood circulation. This is a light microscopic picture to transverse section in the pancreas, showing that the pancreas divided by the trabeculae into lobes or ill-distinct lobes and lobules. And the parenchymatous part of pancreas formed of exocrine part and endocrine part. Exocrine part form of pancreatic acini, which release the pancreatic enzymes directly in the ducts to reach these enzymes to the duodenum. And the endocrine part, which are called the islets of Langerhans. This is a light microscopic picture to islets of Langerhans, showing that these islets formed of branching cords of cells separated by fenestrated blood capillaries. Formed of group of cells separated by fenestrated blood capillaries, as its endocrine part secrete hormones directly into the blood. Islets of Langerhans contain many types of cells in islets of Langerhans, which are A cells or alpha cells, which form about 20%. B cells or beta cells, which form 70% of islets. D cells or delta cells form about 2 to 5%. And the other few cells called F cells, G cells, autonomic ganglion cells. So, the islets of Langerhans, or the main three types of cells in islets of Langerhans, are alpha cells, the main type of cell called beta cells, and delta cells. Then other few cells called F cells, G cells, and autonomic ganglion cells. This diagram also illustrates islets of Langerhans, which is scattered between pancreatic acini, the exocrine part of pancreas, and islets of Langerhans, the endocrine part of pancreas. Islets of Langerhans contain the following cells, which are not appear by hematoxin and eosin. These cells need special stain to can differentiate between each other. These cells are alpha cells, about 20%. Beta cells. Beta cells are the main cells in islets of Langerhans, which form about 70%, and delta cells, which form about 2 to 5%. Then F cells, G cells, autonomic nervous cells, which control the islets of Langerhans, give autonomic control to islets of Langerhans. Fenestrated blood capillaries present between the cells as its endocrine part to secrete its hormones directly inside these fenestrated blood capillaries to reach blood circulation. Alpha cells or A cells, their percent about 20%. There is inverse relationship between the percent of these cells and their size. The percent is 20%, so these cells are larger in size than beta cells. These cells present in the periphery of islets of Langerhans. Present in the periphery. About 20% present in the periphery. They are polygonal cells with central rounded nuclei. Polygonal cells with central rounded nuclei. Cytoplasm of alpha cells. As we mentioned before, all cells in islets of Langerhans or islets of Langerhans appear as pale areas scattered between darkly stained pancreatic acini. So all cells of islets of Langerhans appear pale by light microscope, pale by hematoxylin and UC. By electron microscope, these cells secrete glucagon hormone, which is protein in nature. So the EM picture of these cells is the EM of protein secreting cells that contain rough endoplasmic reticulum, well-developed Golgi apparatus, and membrane-bound secretory granules. But these granules, the granules of these cells, take this shape, specific shape. These granules give dense homogeneous core surrounded by electron lucent halo. Dense homogeneous core surrounded by electron lucent halo. These granules, which contain the secretion of these cells, which is glucagon hormone. This glucagon hormone increase the blood sugar level. So alpha cells secrete glucagon hormone, which increase blood sugar level. Beta cells or B cells, which are the main cells in islets of Langerhans, is its percent about 70%. They lie in the center of islets of Langerhans. This is inverse to the alpha cells, which lie in the periphery. Beta cells lie in the center, and it's a percent about 70%, so they are small in size, from 10 to 15 micrometer. As we mentioned, there is inverse relationship between the percent of cells in islets of Langerhans and their size. Beta cells 70%, so they are small in size, about 10 to 15 micrometer. Shape of beta cells, they are oval in shape with central rounded nucleus. Cytoplasm of beta cells or B cells. By light microscope, these cells are pale basophilic with hematoxylin UC. This is the reverse of alpha cells, which appear pale acidophilic with hematoxin and eosin. But beta cells pale basophilic with hematoxin and eosin. Beta cells secrete hormone called insulin hormone. This hormone, protein in nature, so the EM picture of beta cells is the same as protein secreting cells. What is the EM picture of or electron microscopic picture of protein secreting cells? Protein secreting cells must contain a well-developed rough endoplasmic reticulum, well-developed Golgi apparatus, and membrane-bound secretory granules that contain the protein released from the Golgi apparatus. The granules is different from cells to cells. As we mentioned, the granules of alpha cells were with homogeneous electron dense core surrounded by electron lucent halo. But the granules of beta cells, as you see, the granules of beta cells has crystalloid core. This as a crystals, so its core crystalloids, surrounded by electron lucent halo. Again, beta cells, the electron microscopic picture of beta cells as protein secreting cells contain well-developed rough endoplasmic reticulum, well-developed Golgi, and membrane-bound secretory granules. These granules have crystalloid core surrounded by electron lucent halo. Beta cells secrete hormone called insulin hormone. Insulin hormone is the main hormone in islets of Langerhans. Insulin hormone responsible for lowering of blood glucose level. We mentioned before that alpha cells secrete glucagon, which increase blood sugar level, but insulin lower blood sugar level, but glucagon, which is secreted from alpha cells, increase blood sugar level. As you know that pancreas contain 1 million of islets of Langerhans concentrated at the tail region of pancreas. So when we take section in the tail of pancreas, we will find exocrine part which formed of the pancreatic acini that secrete pancreatic enzymes or digestive enzymes in these ducts to reach to the duodenum and help in digestion. And endocrine part, which form mainly from alpha cells and beta cells. Alpha cells are pale acidophilic cells located in the periphery and about 20%, and beta cells are pale basophilic about 70% and located in the center. This is a higher magnification to the beta cells. Illustrates that beta cells secrete the main hormone of islets of Langerhans, which is the insulin hormone. Insulin hormone lowering the blood glucose level. By help in the process called glycogenesis from glucose. So insulin lower blood glucose level. We studied that the main cells in the islets of Langerhans are the alpha cells and beta cells. So it's very important to know this comparison between alpha cells and beta cells of islets of Langerhans. The titles of comparison are the number, site and size, shape, and the granules inside the cells, and their function. Alpha cells. Number of alpha cells, 20%. It's few in number. Beta cell, about 70%. It's numerous in number. Alpha cells located at the periphery of islets of Langerhans, and they are large in size. But beta cells located in the center, and they are small in size. As we mentioned, there is inverse relationship between the percent and the size. Alpha cells 20% large in size, but beta cells 70% but small in size. Shape of the two cells are the same. Alpha cell and beta cell polygonal with central rounded nucleus, as they are protein secreting cells. Polygonal in shape with central rounded vesicular nucleus or active nucleus, as the two cells are protein secreting cells. The granules inside alpha cells are dense homogeneous, surrounded by a narrow electron lucent halo. Dense homogeneous in the center, surrounded by electron lucent halo. But granules in beta cells has crystalloid core surrounded by white electron lucent halo. This is narrow halo and this is wide halo. Alpha cells, the granules in alpha cells stain red with Mallory stain. Azan stain. But beta cells stain brown with Mallory Azan stain. This brown and this red. This is the stain of granules inside the cells. The granules in alpha cells are alcohol resistant but water soluble. But the granules in beta cells are the reverse, alcohol soluble but water resistant. The function of alpha cells is the secretion of glucagon hormone, which increase the blood sugar level, cause hyperglycemia. But beta cells secrete insulin hormone. Insulin hormone lowers the blood sugar level and cause hypoglycemia. So it's very important to know the difference between alpha cells and beta cells, which are the main cells of islets of Langerhans. Delta cells or D cells. These cells are the third type of cells in islets of Langerhans. These cells are 5% and they are located at the periphery of islets of Langerhans as alpha cells. Delta cells are the largest cell in islet of Langerhans. They are oval in shape with central rounded nucleus. Oval with central rounded vesicular nucleus, as these cells are protein secreting cells, so its nucleus is active nucleus, central rounded vesicular nucleus. The cytoplasm of delta cells not seen by hematoxin As we mentioned, alpha cells appear acidophilic, pale acidophilic with hematoxin and leucine. Beta cells appear pale basophilic with hematoxin and leucine. But delta cells not stained by hematoxin and leucine. These cells only stain with silver stain. Only stain with silver stain. So they not appear by hematoxin and leucine. The electron microscopic picture of delta cells is any protein secreting cells. As these cells secrete somatostatin, they secrete somatostatin, which is protein in nature. So these cells showing the EM picture of protein secreting cells, which is well-developed rough endoplasmic reticulum, well-developed Golgi apparatus, and many secretory granules or membrane-bounded granules that contain the secretion. The granules in the delta cells are larger and less electron dense than that of alpha and beta cells. Delta cells secrete secrete somatostatin. Delta cells secrete somatostatin. Somatostatin controls the release of insulin and glucagon. This somatostatin controls the secretion of alpha cells, which is glucagon, and that of beta cells, which is insulin. So delta cell controls the secretion of alpha and beta cells. F cells are a few cells present in islets of Langerhans. They are few in number, about 1%. They are also protein secreting cells. Their granules are few in number. These cells secrete pancreatic polypeptide, which is called the PP. This pancreatic polypeptide lowering the pancreatic enzyme secretion from the exocrine part. So these cells control the secretion of the exocrine part, as these cells secrete PP or pancreatic polypeptide. This pancreatic polypeptide lowering the secretion or inhibit the secretion of the reduce the secretion of the pancreatic exocrine part or reduce secretion of pancreatic enzymes. G cells are also few in number in islets of Langerhans. G cell, from its name G, secrete gastrin hormone. These gastrin hormone control the gastric motility or GIT motility and increase HCl secretion in the stomach. Ganglion cells are multipolar nerve cells. As any nerve cells, they are multipolar nerve cells. They present also in aggregates in islets of Langerhans. These cells are responsible for the neural control of islets of Langerhans. Islets of Langerhans controlled by autonomic nervous system by these cells. These cells control the autonomic control of islets of Langerhans. They stained by silver stain. Pancreatic islet secretion regulated by two types: paracrine regulation and autonomic regulation. Paracrine regulation and autonomic regulation. Paracrine regulation, the word paracrine means that the secretion of any cell acting on adjacent cell. Secretion of any cells act on adjacent cell. This paracrine regulation occurs in the cells of islets through the gap junctions between the cells. There is gap junction between cells of islets at the following: insulin hormone secreted from beta cells and pass through the gap junction to alpha cells to inhibit glucagon release from alpha cells. Also, glucagon released from alpha cells pass through gap junction to stimulate beta cells to secrete insulin. And delta cells secrete somatostatin that pass through gap junction to beta cells to inhibit insulin and to alpha cells to inhibit glucagon release. So there is paracrine regulation in the pancreatic islet secretion. Another regulation called autonomic regulation. Autonomic regulation occurs through the ganglion cells present between the cells of islets, ganglion cells, which control the autonomic control of the islet. These ganglion cells make sympathetic stimulation to decrease the insulin secretion from beta cells. Again, there is two regulation in pancreatic islet secretion: paracrine regulation and autonomic regulation. Autonomic regulation through the ganglion cells and paracrine regulation through gap junction between the cells of islets. Clinical application. Diabetes mellitus. Diabetes mellitus is a common chronic disease. There are two types of diabetes, type 1 and type 2. Type 1, which is called juvenile diabetes or insulin-dependent diabetes. It's called insulin-dependent as there is autoimmune destruction of the majority of beta cells, as beta cells are responsible for insulin hormone production, which lowers the blood sugar level, so there is defect in the beta cells, and consequently the insulin hormone decreases, then consequently lowering the blood sugar level. So this type treated by insulin injection, as there is defect in insulin amount in the body. But type 2 diabetes, which is called the adult diabetes or metabolic, this is metabolic disorder or non-insulin dependent diabetes, as there is normal beta cell and normal insulin amount in the body, but there is problem in the insulin receptors. Insulin resistance occurs in insulin receptor. There is defect in insulin receptors to respond to insulin hormone. Normal amount of insulin hormone, but there is a problem in the insulin receptors to respond to insulin hormone. So this type treated by diet or other drugs. In the type 2 diabetes, the body isn't able to use insulin in the right way, called insulin resistance, so it's a metabolic disorder. It's very important to know that diabetes, two types, type 1 and type 2. Type 1 called the insulin dependent and occurs early in life, and type 2 called the adult or non-insulin dependent and occurs after 30. Quiz on lecture. An example of a mixed endocrine gland. We mentioned that mixed means that this gland has exocrine part and endocrine part. Suprarenal gland, purely endocrine. Parathyroid, purely endocrine. Thyroid, purely endocrine. The only gland is the pancreas. Pancreas has exocrine part and endocrine part. So the right answer, letter B. The endocrine pancreas is formed of serous acini and ductus. This is the only exocrine part. Cords of cells with fenestrated capillary, so only endocrine. Chromaffin and ganglion cells. C cells, which these cells present in the thyroid follicle. chromaffin and ganglion cells present in the adrenal medulla. Cords of cells with fenestrated capillaries present in the endocrine part of pancreas, which is the islets of Langerhans. So the right answer, letter B. Which of the following cells secrete pancreatic polypeptide, which are called the PP? Alpha cells secrete glucagon, beta cells secrete insulin, delta cells secrete somatostatin, F cells secrete PP, pancreatic polypeptide, which inhibit the pancreatic enzyme secretion. So the right answer, letter D. Which of the following cells inhibit secretion of pancreatic digestive enzyme? Pancreatic digestive enzymes inhibited by pancreatic polypeptide. So the right answer also letter D, F cells, which secrete pancreatic polypeptide. Insulin-dependent diabetes mellitus is due to destruction of which cells? Insulin-dependent, which is called type 1 diabetes or juvenile diabetes, occurs due to destruction of cells that secrete insulin hormone. These cells called beta cells, which is the main cells in the pancreas, in the islets of Langerhans of pancreas. So there is decrease or defect in insulin secretion in this type, though this type treated by insulin injection. The right answer, letter B, beta cells. Which of the following cells inhibit secretion of both alpha and beta cells? Somatostatin released from delta cells inhibits the secretion of alpha and beta cells by paracrine regulation through gap junction between cells of islets of Langerhans. So the right answer, letter C, delta cells. Which of the following cells are present mainly in the center of the islets and their granules have crystalline dense core? Alpha cells present in the periphery of islets. Delta cells also present in the periphery. F cells have few granules. Beta cells, this is the character of beta cells, as beta cells located in the center of islets and their granules also have crystalline dense core surrounded by wide electron lucent halo. So the right answer, letter B, beta cells. The control of secretion from islets is by autonomic and endocrine. No endocrine in islets. Autocrine and endocrine. No autocrine or endocrine in islets. Paracrine and endocrine. Also no endocrine. Autonomic and paracrine. Yes, there is autonomic control by the ganglion cells and paracrine control through the gap junctions between cells and each other. So the right answer, letter D, autonomic and paracrine regulation to the secretion of islets. We have finished now the last lecture of endocrine system. Thank you. Assalamu alaikum wa rahmatullahi wa barakatuh.